Afternoon, everybody. Sepsis is one of the most important unmet medical needs facing humankind today. In any given year, there are 18 million cases on Earth. 6 million of those people will die. In other words, you've got a 1 in 8 chance of dying of sepsis. Sepsis is caused by the overstimulation of the immune system, usually in response to an infection that has entered the bloodstream. One of the key factors that triggers this immune response is a blood protein called C5a. C5a in humans is normally present and an important part of your immune system, but when it becomes out of control, it turns on a massive cellular and cytokine immune response that can lead to multi-organ failure and death. At the University of Limerick, the research team led by Jackie Cooney and Todd Kagawa and myself have developed an extracorporeal device. This is a cassette that is used in plasmapheresis, a form of dialysis in the intensive care unit. It is based on patented immobilized enzyme technology from the University of Limerick, and it specifically and directly targets just C5a in blood. In collaboration with clinical colleagues at Lund University Hospital in Sweden, we have monitored patient C5a blood levels during sepsis, and in our in vitro sepsis simulation studies, we've been able to show that our cassette is able to reduce C5a at levels comparable to those seen in even the most severe sepsis cases. As well as being a very important medical need, there is an enormous market potential for this product as well. Currently, treatment for sepsis involves antibiotics, IV, and fluids, along with long-term stays in intensive care units. In the US, for example, the average length of stay is 25 days in an ICU for a sepsis patient. The cost to healthcare is enormous. In the US alone, $25 billion a year is spent treating over 1 million sepsis patients. The market for a new product that would reduce this is considered to be at least $10 billion just in the EU and US. Customers for such a device are the critical care units of hospitals. Our plan as Cala Medical is to bring this device to a fully approved state in the EU initially, followed by the US, and ideally with a preferred partner which would have the infrastructure in place already to market and sell this directly to the ICU staff who would be treating sepsis patients. We have a strong and dedicated team in Cala Medical. I have over 25 years of R&D experience, predominantly in the United States pharmaceutical industry, working on research projects across the US, Europe, Asia, and Australia. Dr. Cooney has worked, she's a senior lecturer at the Department of Life Sciences in UL, and has over 30 years of protein characterization, expression, and protein mobilization, and has worked in Canada, New Zealand, Ireland, Sweden. Our third co-founder, Dr. Todd Kagawa, is from Hawaii. He has 30 years experience in protein structure and crystallization and has also worked in New Zealand, Oregon, and as well as here in Ireland. We are seeking an initial investment of $1.5 million to give us an 18-month window to scale up our prototype device to GMP level in preparation for clinical trials in humans, as well as gaining regulatory approval in Europe initially, followed by the US. A new sepsis therapy is against a clinically validated target is a massive medical breakthrough, we believe, potentially a game changer in this field, as well as a $10 billion market for those who deliver it. Thank you.